Now, human beings are diploid organisms and hence possess only 2 of the 3 alleles or the gene I. Consequently, as you can see, there are 6 possible combinations of alleles. Therefore, if the allele from one of the parents is capital IA and from the other is small I, only capital IA is expressed in the offspring, as small I does not produce any sugar. In this case, the RBCs have only type A sugar in them. And the genotype of the offspring is capital IA small I, and the phenotype or the blood group is A. Similarly, when the allele from one of the parents is capital IB and from the other is small I, only capital IB is expressed in the offspring, as the RBCs have only type B sugar in them. In this case, the genotype is capital IB small I, and the phenotype is B. However, when the allele from one parent is capital IA and from the other is capital IB, both the alleles express their own types of sugar in the offspring due to codominance. In this case, the RBCs have both A and B types of sugars in them, and the genotype of the offspring is capital IA, capital IB, and the phenotype or the blood group is AB. Now, if the alleles from both parents are capital IA, once again the RBCs have only type A sugar in them, and therefore the offspring has a genotype capital IA, and phenotype A. Likewise, if the alleles from both parents are capital IB, the RBCs have only type B sugar, and so the offspring has a genotype capital IB, and phenotype B. However, if the alleles from both parents are small I, the RBCs have no sugar in them, and so the genotype of the offspring is small I, and the phenotype is O. Therefore, as there are 3 different types of alleles, there are 6 possible genotypes and 4 different blood groups or phenotypes that exist. Apart from being a good example for codominance, the ABO blood grouping system is also an ideal example of multiple alleles. As we just saw, the gene I has 3 alleles, capital IA, capital IB, and small I, that govern the same character, that is, blood group. However, since an individual can have only 2 alleles, it is possible to learn about multiple alleles only when detailed population studies are conducted. According to the multiple alleles concept, more than 2 alleles govern the same character. Now, let us study an experiment that shows how a single gene may sometimes produce more than one effect. Pea seeds have a single gene with 2 alleles, capital B and small b, that control starch synthesis in them. In the experiment conducted, effective starch synthesis was seen in capital B homozygotes, and the seeds produced were large or had a large amount of starch in them. Moreover, the pea seeds were round. On the other hand, small b homozygotes were not very efficient at synthesizing starch, and so the pea seeds produced were small or had less starch in them. Also, the seeds were wrinkled. Moreover, in the experiment conducted, the seeds or the starch grains produced by heterozygotes capital B were of intermediate size, but were round. Now, in this experiment, if we consider the starch size as the phenotype, then the alleles are displaying incomplete dominance. On the other hand, if we consider the shape of the seed as the phenotype, then the alleles are displaying dominance, as the seeds were round like one of the parents. Hence, the experiment shows that dominance is not an autonomous feature of a gene or gene product. Therefore, dominance, incomplete dominance, or codominance depends on gene product and the production of a particular phenotype from this product. It also depends on the phenotype that we wish to examine in case the same gene influences more than one phenotype. Did you know that Gregor Mendel cultivated and tested about 29,000 pea plants between 1856 and 1863? One such test was the crossing of pea plants with yellow and round seeds and those with green and wrinkled seeds. As you know, such a cross between plants that differ in two traits or characters is known as a dihybrid cross. This experiment, conducted by Mendel, resulted in a pea plant that produced yellow and round seeds in the F1 generation. Here, can you guess the dominant shape and color? Obviously, the round shape is dominant over the wrinkled shape and the yellow color dominant over green. Mendel noticed that these results were identical to the results he had obtained when he had carried out monohybrid crosses between plants with round and wrinkled seeds and those with yellow and green seeds. Now, in Mendel's dihybrid experiment, let's denote the dominant yellow seed color with the genotypic symbol capital Y and the recessive green seed color with small y. Likewise, let's denote the round seed as capital R and the wrinkled seed as small r. The genotype of the parents can therefore be denoted as capital R, capital Y, and small r, small y, small y. On crossing the plants, the gametes capital R, capital Y and small r, small y unite to produce yellow and round seeds with the genotype capital R, capital Y, small y in the F1 generation. Mendel further self-pollinated the plants obtained in the F1 generation to produce F2 generation plants. This time, he found that 3/4 of the seeds were yellow, while 1/4 was green. Similarly, he noticed that 3/4 of the seeds were round, while 1/4 was wrinkled. Therefore, the yellow and green color had segregated in the ratio 3:1, similar to the results obtained in a monohybrid cross. And the round and wrinkled shape had segregated in the same ratio 3:1. Mendel also found that the phenotypic ratio in the F2 hybrid was 9:3:3:1. That is, there were 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, and 1 wrinkled green seed. Mendel had got the same phenotypic ratio in several dihybrid experiments that he had conducted. This ratio 9:3:3:1 can be derived as a combination series of 3 yellow:1 green with 3 round:1 wrinkled. And further, this derivation can be written as shown. From such observations on dihybrid crosses, Mendel proposed a new set of generalizations, which was later called the law of independent assortment. According to this law, when 2 pairs of traits or characters are united in a hybridization experiment, the segregation of one pair of characters is independent of the other pair of characters. We can use the Punnett square to easily understand the independent segregation of the 2 genes during meiosis and the production of pollen grains and eggs in the F1 generation. For example, In the dihybrid cross experiment conducted by Mendel, the genotype of the seed obtained in the F1 generation was capital R, small r, capital Y, small y. Now let's consider the segregation of one pair of genes, capital R and small r. Here, 50% of the gametes have the capital R gene, and the remaining 50% have the small r gene. Now, apart from each gamete having either capital R or small r, it must also have either capital Y or small y. Here, it is important to note that the segregation of 50% capital R and 50% small r is different from the segregation of 50% capital Y and 50% small y. Hence, 50% of the gametes bearing small r has capital Y, and the remaining 50% has small y. Similarly, 50% of the gametes bearing capital R has capital Y, and the remaining 50% has small y. Therefore, the genotypes of the gametes are of four types, namely capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. In other words, there are four types of eggs and four types of pollen. And as you can see, the frequency of these gametes is 25% or 1/4 of the total number of gametes produced. Now, the allelic composition of the zygotes of the F2 plants can be easily obtained using the Punnett square as shown. As you already saw, there are four different types of phenotypes formed, and the phenotypic ratio is 9:3:3:1. However, 9 different genotypes result from the F2 hybrid, and the genotypic ratio is
1:2:1:4:2:1:2:1.
Thus, from the dihybrid cross experiment, Mendel found that the gene combination of the progeny differs from the parental gene combination. Gregor Mendel, the father of modern genetics, not only researched pea plants, but also carried out several experiments with honeybees. In fact, by 1865, Mendel had published his work on inheritance of characters. However, his work did not receive due recognition until the beginning of the 20th century due to several reasons. Firstly, Mendel's work couldn't be widely publicized because means of communication in those days were not well developed as they are today. Secondly, Mendel had considered genes or factors as discrete and stable units responsible for controlling the expression of traits. He had also explained the concept of alleles and how they were responsible for the continuous variations observed in nature. However, his concepts about genes were not readily accepted by his colleagues. Thirdly, Mendel, for the first time, had applied statistical analysis and mathematical logic to problems in biology, which was objected to by many biologists of his time. Finally, Mendel could neither explain what genes were made of, nor could he provide any physical proof of their existence. Mendel's work got due credence only in 1900, when his results on the inheritance of characters were independently rediscovered by 3 scientists, Hugo de Vries, Carl Correns, and Erich von Schermach. Also, both chromosomes and genes segregate during the process of gamete formation, and only one of each pair is passed on to a gamete. However, they differ in the way they segregate. In genes, independent pairs segregate independently of each other, while in chromosomes, one pair segregates independently of another pair. Let's now study the behavior of chromosomes during cell division. In the anaphase stage of meiosis I, a set of chromosomes aligns along the metaphase plate. Here, you can see a long orange and a short green chromosome, as well as a long yellow and a short red chromosome aligned at the same pole. Now, consider another set of chromosomes aligned along the metaphase plate during the anaphase stage of meiosis I. Here, there is a long orange and a short red chromosome and a long yellow and a short green chromosome arranged at the same pole. Now, during the anaphase stage of meiosis II, you can see the orange and green chromosomes segregating together in the cell on the left, while the orange and red chromosomes segregate together in the cell on the right. Sutton analyzed this segregation of chromosomes during meiosis and concluded that chromosomes have individuality and they occur in pairs, with members of each pair contributed by each parent. He also concluded that the paired chromosomes separate from each other during meiosis, and the distribution of the paternal and maternal chromosomes in each homologous pair is independent of each other. Both Sutton and Boveri were of the opinion that the pairing and separation of a pair of chromosomes would result in the segregation of a pair of genes or factors carried by them. Their hypothesis on chromosomal behavior came to be known as the Boveri-Sutton chromosome theory. In fact, Sutton combined this hypothesis of chromosomal segregation and Mendelian principles and called it the chromosomal theory of inheritance.